So good morning to all. In last video, I have discussed about general methods for the repropositions of drugs for any disease. In this videos, I am going to discuss particularly about the repropositions of drugs against COVID-19. So before the repropositions, I am just going to summarize about the structure of this virus. First, we have to understand what this virus contains. So this virus is a single strand positive sense RNA virus, and it contains various proteins like envelope proteins, there, membrane proteins. The proteins, particularly spike protein, this proteins helps in the entry of the virus inside the host cells. There are another materials of the virus, RNAs, and then nucleic acid protein. So, in repropositions of the drugs, means we are exploring the existing molecules for the treatment of particular disease. So, one approach you can think of to target the specific targets of the virus, you can target about the spike proteins. You can repurpose a molecule which will inhibit the envelope glycoproteins, which will inhibit nucleic acid proteins, or any other target which is viral specific. So what in case of virus, as virus enter inside the cells and use the host machinery for the replication, so you can also repurpose, or you can also think of the molecules which can inhibit the host targets or host machinery which virus is using for the replication. So this is a two way, particularly in the microorganisms, we can target. One is target where you are targeting the specific structures or specific targets which is not available in the host, and another is you are targeting the host. So in COVID-19, this is the one summarized pictures where I am going to tell you about repurposing of the drugs which are repurposed for the targeting SARS-CoV-2. So this is the SARS-CoV-2. As I told you, there is a spike protein particularly is helps in the entry of this virus inside the host cells. If you see these pictures, so there is a TMP-RS2, and TMP-RS2 is a transmembrane protease serine 2. This is a host protease. This host protease, what it will do, it's in the lungs, and it do the breakdown of spike protein into two forms, S1 and S2. S1 will attach to the AC2. This is also another host enzyme which is present in the lungs, and S2 will attach to TMP-RS2. This is the transmembrane protease serine 2. After attachments, it will enter, the endocytosis process will happen, and endosome formations and endosomal acidifications, which is required for the proper pathogenicities of this virus. And but after that, it will release the RNA, and the released RNAs with the help of proteases or proteolysis will happen, which will cleave the specific proteins into a not specific protein. It will cleave the peptides into proteins, specific proteins, and do the formations of replication complex, including RNA dependent RNA polymerases. And these all replication complexes will finally results into transcriptions, and then translational, and then finally nucleic acid, spike, envelope, or specific proteins will be formed. And these nucleic acid, spike, and envelope proteins finally results in the formations of new virions, and at last exocytosis of new virions will happen, and new that one virus replication process will happen. This is the simple cycles by which this COVID-19 to enter after entering the inside the host cell, how it replicate. So for the repropositions of drugs, you can inhibit. First promising approach is you can inhibit if you have any molecule which will inhibit the entry of the virus inside the host cells. Either you should have some spike inhibitors. Research is going on for the new molecules for the inhibitions of spike proteins. As in market, spike protein, specific COVID-19 spike protein inhibitors are not available. So another thing is what we have in the market, TMP-RS2 inhibitors we have, like nafamostat or chemostat. Nafamostat in the market, it's available as a short-acting anticoagulant and approved in the Japan. Chemostat is another molecule which is used for the treatment of chronic pancreatitis or reflux esophagitis. So these molecules are actually available in the market in worldwide. Not in India. I don't know exactly about the status of these molecule in India, but these are available, approved in the Japan, and these molecules have ability to inhibit the TMPRS2. So these molecules can be repurposed for the treatment of COVID-19 too. So it's under investigations whether these will get a benefit or not. Many of you have heard about chloroquine. Chloroquine is a antimalarial molecules which inhibit the AC2 or another actions of hydroxychloroquine or chloroquine is they increase the basic environment of the endosomes. So as basic environment is increased, the proper pathogenicity of virus will not happen. As you know, the endosomal acidification environment is required for the proper pathogenicities of this virus. So another molecule, imatinib is a get approved by the Novartis for the chronic myeloid leukemia. It's a kinase inhibitor, so it could inhibit the endocytosis of the endocytosis process. So it could also be repurposed, and research is under investigation. Another molecules, these lopinavir or ritonavir, you have heard, these are the protease inhibitors available in the market for the treatment of HIV virus. So it can inhibit particularly 3CL proteases. Disulfiram is also the drug which is available in the market for the alcohols, and it can inhibit the PLPRO. So ultimately specific proteins will not form. Another drug, remdesivir. Remdesivir, you might have heard number of news regarding remdesivir. Remdesivir is particularly the drugs which is discovered for a Ebola virus, tested in the Ebola virus but not find effective, and later on it's find effective against MERS virus or SARS-CoV-2, and these two viruses are similar to COVID-19. So these drugs might have more chances to develop as a anti-COVID drug. Another drug is favipiravir. It's a drug used in the influenza. It's available in the market, particularly in the Japan, for the treatment of influenza. It's also have the ability to inhibit the RNA-dependent RNA polymerase. Cyclosporin is another drug. This is the immunosuppressant, particularly used in the treatment of cirrhosis, Crohn's disease, and any others autoimmune disorders. So it can inhibit this transcriptions process. So ultimately the particular proteins will not form and virus replications will not happen. IF and beta is another drugs which are under investigations. There is a serlimumab or tolizumab. These are the specific monoclonal antibodies against IL-6 receptors and used in the treatment of rheumatoid arthritis. So it can also used as a repurposed as in the treatment of SARS-CoV-19. So these are the few examples of the drugs which can be repurposed for the treatment of COVID-19 too. There are a number of drugs which are under investigations or repurposing. There are various databases also available online, so you can get this COVID-19 repurposing database. You have to just add your institutional ID or your organizational IDs, and you will get the information, and you will find more than 100 molecules are under repurpositions. Some molecules are under preclinical studies, some are in clinical trials. So the question is how much these molecules are promising, means these repurposed molecules. So repurposing, if you see the recent articles which are published in the Lancet, one of the reputed medical journals, you will find that they have recently retracted the paper on hydroxychloroquine, which have earlier accepted for the publications, and they have claimed that hydroxychloroquine or chloroquines can useful for the treatment of COVID-19. But finally they are unable to prove whether it is helpful or not, and ultimately this paper has been retracted. If you see, this is a recent papers on remdesivir, and if you see the results of this paper, just go through this paper and go through the results, you will find that author has concluded remdesivir they have tested, they have found the results but not significant. And further, the testing of these drugs in a large number of populations or large number of subject is required to make any conclusions. So means repurposing is a good options, as it's saving the time and money which is involved in the drug development process, but same times it's also challenging. It's not that much easy. You can easily put the molecule for the market, as you have, many of you might be heard about hydroxychloroquines itself leading to a death in some comorbid patients. So very careful. Challenges in the reproposition: first challenges is aggregations, because you have to aggregate search and analysis, and there is a vast amount of data which is captured. So you have to be make efficient and effective use against a new indication. It's a biggest challenge. That's why nowadays you have heard about artificial intelligence or machine learning might play a key role during these drug repropositions or in drug industries. Another challenge is the safety concern. If suppose the molecule which you are repurposing is showing efficacy but at higher concentration at which, or at a higher strength at which it is available in the market, so obviously the question will raise on its safety. So you have to check the safety first. Another things, if you are repurposing a molecule and molecule earlier is available for suppose diabetes conditions, now you are repurposing that molecule for the COVID-19. So there might be that molecule may be safe or may not be safe in when you are switch over to the indications, because disease biology, every disease biology is different. So another challenge in the drug repurposing is IPR issue. As you know, we are repurposing the existing molecules, and existing molecules are already patented by someone. So there is a number of debate is going on, particularly in India. We can't we can't patent a repurposed molecules. So these are the IPR issue, and if we are not able to patent, if companies are not able to patent it, then they will not get that much benefit. These are some issues, challenges. So future perspective, yeah, as you know, but this is my personal observations, as these viruses, now it's COVID-19 is doing all these things. Maybe after 1 year or maybe 10 years or 20 years, new virus will come. So these viruses are evolving year by year. If you have heard recent news, Hanta virus is another virus which are emerged in China, and it can also spread. But fortunately, this virus is not that much pathogenic or not spreading that fast as the COVID-19. But viruses or all these things can be evolved time to time. So we have to learn how to live with these viruses. We have to take the precautions. And future, I hope a lot of research is going on vaccines, and I hope these vaccines will soon come into the market. Vaccine is the most promising approach where you can protect the populations from the effect of these type of viruses. So another thing is, I just added 1 or 2 slides what you can do as a students, particularly for the students. So as due to lockdown, everyone is in home or getting frustrated also, because we are not used to that much to spend much more time at the home. So you have to read the literatures and think about it, generate your ideas and share your ideas with the governments or through to pharmaceutical companies. And I believe the true sign of intelligence is not in knowledge, it's imaginations, and for imaginations you can do from a home also. This recently MHRD drug discovery hackathon is going to release there all of you can participate, give your ideas how to tackle with the problems of COVID-19. You can do spread. These are some picture I have added. You can see these are all the Indian traditions which protect us from many type of these virus infections. reactions. So we have to work together, computational scientists, biologists, pharmacists, doctors, everyone's we